Well, it's a pleasure to be introducing our next speaker today. Joseph has been with me since the beginning, it feels like. He did his bachelor's degree at Texas Tech, then his master's degree also at Texas Tech here in Lubbock. And then finishing that in 2018, he started his Ph.D. at A&M with Dr. Julie Howe and I. And today, Joseph will also be talking about cover crops, but he will be discussing what the effects of cover crops are in cotton production systems, specifically in our harsh semi-arid environment that we have in the southern high plains of Texas. With that, Joseph, I'll turn it over to you.
Yes, of course. Thank you, Dr. Lewis. I heard there was a problem with my screen share. How about that? Is that better?
Looks good.
Okay. All right. Well, thank you for that introduction, Dr. Lewis. My name is Joseph Burke. If I haven't had the pleasure of meeting you, hopefully I'll get to see you at the Tri-Society meetings starting in 2 weeks. Again, my work is focusing on both no-tillage and cover crops in these semi-arid cotton production systems. And the reason why we have those two combined is because the way the study is designed, we can't separate those interactive effects. But in the high plains, you wouldn't have no-tillage without a cover crop in most instances. So it makes sense to study those coupled together. Also, I want to appreciate and offer a thanks to Jack for doing a great job of introducing cover crops. And we're going to talk about these in a little bit of a drier environment out here in West Texas today. So I want to start by saying that I wish you were all here in Lubbock so we could take a trip outside, walk through the wind, and then walk back inside so I can highlight why exactly we need cover crops in the first place. If you can get to my next slide. We can't talk about cover crops in West Texas without talking about where they come from. And the first place to start with that is the Dust Bowl. So beginning back in 1935, this is the quintessential picture that we see in places, focusing on just how bad the Dust Bowl was in West Texas. However, if you go outside, you still see it all the time. So this picture was taken in 2013 in Terry County by Dr. Lewis's mother-in-law, or soon-to-be mother-in-law at the time, highlighting just how bad the dust storms can still be in those dry years. This picture was taken in 2019, just outside of the research center here in Lubbock, looking at a pretty bad dust storm that was coming in May of that year. And then finally, this one was taken in February of 2021. And this was me trying to drive home from the research center that day. So this is about 5:30 in the afternoon. So just highlighting just how bad our dust storms can still be. Now with that, I will say we have reduced wind erosion on the Texas High Plains by about 80% since the 1930s. So we've done some great work, but there's still some work to do. And one of the things that makes that challenging is our environment. It's pretty warm here, between 13 and 17 degrees Celsius. We get anywhere between 405 and 560 millimeters of rainfall a year. So that's about half of what you receive in College Station. And our wind speeds are about 19.8 miles or kilometers per hour. So really fast wind speeds. We're the fourth windiest place in North America. And we have frost-free days of about 195 to 255 days. So what's important to note is that, starting around May or June, when we're going to put those cotton plants in the ground, is when our temperatures and our precipitation are highest, but also our wind speed is one of the highest that we can receive, which causes some pretty negative impacts when it comes to planting at those times. And finally, something to note about West Texas is that our potential evapotranspiration is 2 to 3 times what our annual precipitation is. So it makes for a really harsh environment to work in. Then you couple that with the soils that we have. And then in this area, we're going to focus on MLRA 77C, which is the Southern High Plains. And it's a pretty big area of Texas, and it's formed by Aeolian deposits during the Pleistocene age. And the predominant soil orders are the alphasols, inceptisols, mollisols, and vertisols. But today I'm going to talk about Paleostaus, specifically the Amarillo soil where I've done all of my work. And for the focus today, I'm going to talk about 2 sites that we worked with. The primary site is going to be our cropping systems location. And this is the Agricultural Complex for Advanced Research and Extension Systems in Lamesa, or Ag Cares as we call it. And about 22 miles as a crow flies, we can get to our native site, which is our typical comparison site. So if we hadn't broken it out, if we hadn't plowed, if we had just let native grasses exist, what would the potential be for carbon sequestration or carbon storage? And so that site is located near Wellman, Texas. And I know 22 miles is pretty far to make a comparison, but that is the closest native range site in the area. There's not a lot of those that exist. But the nice thing about both of these is they're both Amarillo fine sandy loams with pretty similar slopes of about 0.5 to 1%. And defining that soil is, again, a Palais Oosthouf. It's 80% sand, 9% silt, and 11% clay in Lamesa. There's a CEC of about 10 centimoles charge per kilogram of soil, and it has an average pH in the region of 7.8. However, which is important to note, our cover crop plots, the pH is 7.2 with a reduction that we've seen over the last 22 years of the study. And then soil organic carbon is around 2 grams per kilogram. So really small amounts of soil organic carbon. What's interesting to note, though, is this is an Amarillo fine sandy loam, which means that we can kind of extrapolate the results that we're going to see with the study to other cropping regions or other cotton cropping systems in the region. Looking at our site specifically, again, this is at the Agricultural Complex for Advanced Research and Extension Systems in Lamesa, and we were looking at 3 primary cropping systems. The first 2 are continuous cotton systems were established in 1998 by Drs. Keeling and Bronson, when Dr. Bronson was still here. And they looked at a conventional tillage winter fallow, which is our traditional system that we see on the Texas High Plains, compared back to a no-tillage with a rye cover crop. And that rye was planted at about 33.6 kilograms per hectare. That is based off an NRCS recommendation. And so then in 2014, a grant that was received by Drs. DeLon, Keeling, Lewis, and I believe Dr. Foster was also on that one. They split those plots in half, those no-till rye cover plots, and they added a mixed species cover. So we went from 26 row plots, or I'm sorry, 32 row plots to 16 row plots. And in that mixed species cover, we have 50% rye, 33% Austrian winter pea, 10% vetch, and 7% radish by weight. I'll note before we get too deep into the presentation that because of the harsh winters we've had, when we speciate out that cover crop biomass at the end of the growing season in March, we have about 99.8% of that is the rye in the mixed species. So how I like to describe these systems is we have a rye and we have a very expensive rye cover crop system. And again, we compared that back to a native rangeland. It's a native system in Wellman. We don't have historical records that are now 90 years old, but those 90-year-old records show that it was never plowed, and we assume that it wasn't plowed before that. It's a randomized complete block design, and the soil samples I'm going to talk about today were collected at 0 to 5, 5 to 10, 10 to 30, 30 to 75, and 75 to 100 centimeters. That's broken up by the soil horizons. These are big boy plots. These are 16 rows by 76 meters long. So they're pretty big plots there in Lamesa. All right, and then looking at the timeline, one of the reasons why it's difficult to grow cover crops here is because we have a pretty narrow window from once we get that cotton crop out of the ground to when we can go back and plant our cover crop into it. So the top bar or the top line shows our traditional cotton agronomy timeline with a fallow period from November to early May. And then we're going to have cotton growing from mid-May through November. And then when we harvest, we obviously will come back in and there'll be several plowing events in the spring. So when we insert our cover crop into that system, we have to get it in almost immediately after we plant our cotton. And then we're going to terminate it early enough out to where we can recharge some of the water that was used by that cover crop prior to planting our cotton crop. Because if you remember back to our climate slide, we get most of our water in early May. And so we want to make sure we can maximize on that recharge event by having some of that cover crop biomass there, but that's not actively growing, taking up that water that will be used for the cotton crop. The pictures here highlight during the growing season what that cover may look like throughout the season. You can see that we can generate quite a bit of biomass in our pretty semi-arid system here. And with that, I want to show just how much biomass we have been able to produce in dry matter in kilograms per hectare. So I joined the study in 2016, but they started the study collecting a biomass in 2015. So from 2015 to 2020, this is the amount of biomass that we've been able to generate. What's interesting is in the odd years, we've seen a significant difference between our two treatments with our no-till rye cover crop systems generally producing greater biomass compared to our no-till systems with the mixed species cover when we do have differences. However, in 2019, we did have greater biomass production in the no-till mixed species. Reason for this was we ended up getting a bad batch of rye for our single species cover, and so we just didn't get a lot of biomass germination in that year with the single species cover. The question earlier was talking about soil organic carbon. This has been one of the focuses of my dissertation. And looking at the four systems across the x-axis, we have our continuous cropping system with cotton, with traditional fallow and tillage, compared to a no-till with the mixed species, no-till with a rye, and then finding our native rangeland. And we looked at the carbon stocks throughout the depth of the profile until we got to 100 centimeters, which is about the point where we always hit caliche. So this is going to showcase just how much organic carbon we can build within our system when looking at us as a stock. Now, one thing to note is this isn't just a concentration. This is the concentration multiplied by the bulk density multiplied by the depth interval. And so that's why we see so much organic carbon in those lower depths, is because it's just based on the size of those depths as well. But if you look at the profiles, what we see are there's significant differences in our top two depths and finally at our bottom depth. And in those top two depths, we see that there's no significant difference in soil organic carbon between both of our conservation management practices and our native systems, showcasing that we can actually sequester about as much carbon in those two depths as we can in our native system when you compare that back to our conventional tilled system. However, if you look at the bottom depth, we do see significantly greater soil organic carbon in that native system compared to all of our other treatments. One of the reasons why we think this is just because our native systems have deeper rooting grasses that are perennials. And so they can actually have roots that penetrate deeper into the soil profile, building up that soil organic carbon over time. And one of the things that we really like to do in our lab group is showcase the relative changes in soil organic carbon compared back to that conventional system. So how much carbon can you build over time? And this is 20 years after the study was started. And so we have significantly increased the amount of carbon compared back to our conventional tilled system. So with our two cover crops systems, we've sequestered between 5 and 6 megagrams of carbon per hectare compared back to that conventional system, but still not anywhere close to that native system of around 13 megagrams of carbon per hectare. So we still have a long way to go to get back to our native system, but we are significantly increasing the amount of carbon over time. And this comes out to about 0.1 megagrams of carbon per hectare per year on an average basis, with most of that accrual being later in the study system, not earlier in that study system. One of the other things that we try to focus on is permanganate oxidizable carbon. This is going to be the most active fraction of carbon. So typically we're going to think about the most fresh inputs into the system. And what we can see with our no-till systems with cover crops, we can significantly increase the amount of that active carbon fraction that we're putting into the system compared to both our no-till or our conventionally tilled system and our native rangeland. And one of the reasons we think this is true is because we just have more inputs into the system. We're applying fertilizers, we're applying more water compared to that native system. So we're just increasing the amount of fresh residues that are being cycled within those no-till systems compared back to that native. And that trend continues throughout the depths all the way throughout the profile, except in that middle depth where we didn't see any significant differences. I put the percentages here. These percentages represent the amount of permanganate oxidizable carbon per SOC, so per that soil organic carbon fraction, because epoxy is really just a fraction of that organic carbon, and we want to see how much of that fraction is there. And we can see that we have a significantly greater fraction of the active carbon within our top two depths compared to the rest of the profile, specifically in our no-till systems compared back to that conventional. And that's why we see, that's why we think that it's based on the fresh inputs into the system, because it's giving us more and more of this fraction to add compared back to just the baseline soil organic carbon. I'd be remiss without talking about soil health. This was actually the whole point of the study, was a soil health assessment. So we've done 2 soil health assessments now in the study, one in 2018 and one in 2019. These are the results from 2018, so 20 years of the study. And what we see again is significant increases in both soil organic carbon, enzyme activities, as well as our PLFAs, which is an indicator of microbial biomass, as well as community structure and composition, compared back to that conventional system. However, while our microbial communities are significantly less in our 0 to 5 centimeter depth compared back to our native system with these conservation systems, we do have greater nutrient cycling potential using enzyme assays as a proxy for that. So we do think that it's because of these fresh exudates, the fresh biomass turnover, that we are seeing this biomass cycling and nutrient cycling potential compared back to the conventional system. One of the other things that I hear from farmers all the time when we go to farmer meetings, though, is how does this relate to yield? Is my biology going to influence my yield in this system? And so we compared our biological indicators for soil health back to yield. And when you look at it from a PCA standpoint, there is no relationship between a biological indicator and yield. And we've run this assessment both against averages for yield as well as the individual yield for that year. And part of the problem is that biology is a snap point in time, and yield is a series of events that happens over time. So it's not that I can say that biology hasn't had a significant impact, but the time point that we sampled, it has not had an impact on yield to date. And so that's something important to know as we work forward, is maybe the benefits of cover crops in our system are not necessarily from a biological standpoint, from a physical standpoint about slowing wind erosion and improving water infiltration. And so I saw some questions during Jack's presentation about cotton lint yield. And so I wanted to, or about, production and the variable effects it can have. Over time, we have seen that 2 out of the 6 years that we've evaluated, there has been a significant reduction in yield with these cover crops compared back to a conventionally tilled system. And then in other years, we may not see a significant difference, but there is a numerical difference. And so one thing that farmers are constantly asking is, well, what is causing that yield drag? What is the result of that? And when I first started, I would have said that they're using all the water. And so there's no water left for that cover crop. But over time, I have been less convinced by the water usage theory. And most of my focus now has been put on the nutrient immobilization with that cover crop. So we've collected soil moisture readings bi-weekly for the last 6 years now. And one of the things that we've seen over time with each growing season is with that cover crop, we are going to see a decrease in soil profile moisture. 2019 and 2020 is probably a perfect year to illustrate this, where we see a significant decrease in water prior to terminating that cover crop. However, once you terminate that cover crop, we have significant increases in soil water availability and storage later into the growing season and then throughout the rest of the growing season. So we see this inversion point in most of our years. 2021 has been a weird year because we had a micro downburst about 2 weeks after planting cotton that wiped out the entire crop. And so we don't have the same water usage results in 2021 as we did in 2020 and 2019. But it's nice to see that we see this inversion point in each of these years. So then we went to go look at nitrogen availability to see if there's some immobilization. And like Jack, we used litter bags. Our first set went out in 2018, and we've been doing that every year since then. Jack, I'm not envious of you, though, because we bought our litter bags. I can't imagine having to make them, so I feel for you. And so if you're interested, we buy ours from Bar S in Idaho, and they ship them right to us about 2 weeks after we order them. So if you're going to do this again, I definitely recommend buying them. With our biomass decomposition, we wanted to see just how much nitrogen is being tied up potentially. So in 2020, we produced about 4,500 kilograms of biomass per hectare. When you multiply that by the percent nitrogen in that biomass, you get a potential of between 130 and 145 kilograms of nitrogen per hectare. This is enough for about 2 bale cotton. So we should have plenty of nitrogen for our producers to be able to generate as much biomass or cotton lint as they want to. However, all of that biomass is not going to mineralize at the rate that we need it to be available for the cotton crop. So if you look at this table, this is potentially mineralizable nitrogen in kilograms per hectare. And so if we mineralized half of that, we would have enough for about 1 bale cotton to the acre or to the hectare here in West Texas. So using litter bags, we used fresh biomass so that way we could calculate the amount of nitrogen that's released in these systems for 132 days following termination of that cover crop. And that puts us to about peak nutrient demand uptake in that cotton crop. And what we see is following termination, even 132 days, about 80% of our biomass is still remaining in our systems. So only 20% of that has decomposed, which indicates that we still have quite a bit of that being tied up in our system. So one of the studies that we've started recently is we've been applying nitrogen earlier on in the system to try to see if we can counteract that nitrogen immobilization potential. More on that to come. It's the only chapter I haven't really worked on yet, but it's one of the more exciting ones that we've seen looking at nitrogen immobilization potential. So with that, in summary, I made a figure. Hopefully this represents the ideas well, but this looks at the benefits and the consequences of our conservation cotton cropping systems. So starting from left to right, we do have reduced susceptibility to wind erosion as well as increased infiltration and water storage. We've also seen a decrease in soil pH, which is perfect for our systems where we kind of release some of those micronutrients into our cotton cropping systems. However, we have seen a decrease in yields, which we're still investigating. Part of that may be tied to potential for nitrogen immobilization, but also we've seen an increase in our pathogenic nematode communities following these cover crops, which our plant pathologist is helping to investigate. We also see an increase in carbon storage with our systems, as well as increased extracellular enzyme activities and microbial community size and composition. With that, we also see an increase in water storage as well as decreased evaporation from the soil surface. So there are lots of positives in addition to the few negatives that we see from our cover crops. But like Jack said, it really is dependent on what your system prescribes. I would never recommend that you use cover crops from the way that you use them in College Station as you would in West Texas. And we still need to do a little bit more work to understand how these benefits play into dryland systems, because this is a deficit irrigated system that we're working in. With that, I need to thank my committee members, Drs. Lewis and Howe, my chair and co-chair, as well as Drs. Foster, Gentry, and Boughton. Our collaborators, Dr. Paul DeLon and Wayne Keeling, for all of their support over the years, as well as Texas A&M AgriLife for their support through the Strategic Initiative Assistantship in Water and Soil Health. I'd also like to thank the funding sources for this work, which was AgriLife, the Natural Resources Conservation Service, and Cotton Incorporated. With that, I think I have time for a few questions. Let me know, and I'm happy to discuss any of the work that we've talked about so far today.
Joseph. Can you open your chat window, Joseph?
Yes, sir.
Questions? Questions? Come on.
Well, I'll try one. I don't know if it's a legitimate question or not. You said in your cover crop system that you have a higher concentration of oxidizable carbon. Is that because you versus the native system? Is that because you have this intermediate period where you don't have an active root system and you turn on the necrotrophs that turn over that system, or is it, whereas in the native system you always have the necrotrophs kind of being suppressed? You understand that question?
Yeah, no, so good question. We haven't looked at necrotrophs specifically. So we think that most of that active fraction is just because we're going to have more primary plant productivity in our cover crops during that period earlier in the growing season compared to our native system, which has less net primary productivity. So we have greater exudates and more nutrient cycling compared to that system that doesn't have as much inputs. So we're intensively managing our no-till systems and with our cover crops compared to that native site, which is why we think we have more of that active fraction compared. I should note that we also sampled those soil samples on June 1st and 2nd in 2018. Yes. And that was the hottest day on record for West Texas. Those 2 days that we sampled, it was 108 one day and 109 the other. So where we have those intensively managed systems, we probably have a little bit more soil moisture compared to that native system, which we expect to see a little bit more with that cover crop.
Other questions from our virtual audience? Hey, Joseph, is it sounds like you can only grow cotton and rye there. Is that the case?
It depends on what soil you're in. If you go farther north, you can get some corn in, wheat in. We also grow wheat as a cover crop in some of our other studies. Peanuts are also pretty common along with sorghum. However, in La Mesa, where we're so sandy, the primary crop is going to be cotton.
And so you can't grow a legume there as a cover crop to mitigate the potential effect of nutrient tie-up, eh?
Yeah, so good question. We do try to grow legumes. We just haven't been successful at getting them to do very well yet. So we're also going to investigate one of our cropping systems that we look at is a cotton wheat fallow system. And so we are looking at summer cover crops in that fallow period to try and get some of those legumes to help offset some of the environmental impacts with that. I do have a question here from Dr. Murray. What are you going to do when you graduate? Great question. My mom asks me this all the time too. And so first things first, Dr. Lewis secured a Sustainable Ag Systems grant, and I was lucky enough to be one of the co-authors on the grant. So I'm actually going to stay on and fulfill the obligations that I wrote for a postdoc for her. And so that'll be about 2 years. And then from there, we'll see. I just was accepted as a Encompass Fellow through Bayer Crop Sciences. And so apparently the whole purpose of that is to try to recruit us to work for Bayer. But we'll see. I love academia. I like teaching, but we'll see where the paths go after we finish my component of the SAS grant.
Last chance for questions. All right, that was a great job. Thank you, Joseph.
Thank y'all.
Yeah, thanks also to Jack.